Welcome back. We're moving on in our discussion of simple Mendelian genetics, and our next two recordings are going to look at Mendel's laws. Now, the problem with Mendel's laws is they only apply under pretty specific conditions, and so it's important to know what those conditions are. So Mendel's laws are only really going to work for us when we're dealing with a gene or genes which control, which are responsible for producing a single characteristic. So, for example, pea plant height is under the control of a single gene. So that's called a monogenic characteristic. One gene determines pea plant height. And so there are several of those, 7 or so of those in the case of the pea plants that Mendel and his gardener worked on. As soon as we start getting into polygenic traits, complex characteristics, then things start to become a little bit more complicated. So bear that in mind as we look at Mendel's two laws. We're going to start off with Mendel's first law, which is also commonly known as the law of segregation. And Mendel's first law really showed us, I would say, 3 main things, 2 of which are very, very closely related. You'll do better with Mendel's laws and with genetics in general if you've got a solid grasp of meiosis. So if you're having any problems with the genetics, it could be rooted in a problem with understanding meiosis. So let's take a look at Mendel's law. Mendel's first law here deals with one gene determining a single characteristic, and the gene is going to be the T gene, and it's going to control plant height. And there are 2 variations of the gene, so 2 alleles, and we're going to call those big T, that's an uppercase T, and little t, that's a lowercase t. So big T and little t, those are the 2 alleles of the gene T. And so if you have 2 big T's, you'd be homozygous dominant, and because the big T is dominant to the little t. If you're homozygous dominant for the T gene, then the plant will be tall. Now remember that tall is a trait within the characteristic of height. On the other hand, if you have 2 recessive alleles, like on the right here, then you'll see the effect of those recessive alleles because there's no dominant allele in play, and that plant would be short. Remember, again, short being a trait within the characteristic of height. So it's what Mendel's first law showed us is that alleles of genes segregate during meiosis. And so what that means is that the 2 big T's will separate, the 2 little t's will separate, and they'll end up being placed into gametes. And remember, the gametes will be haploid, so the sperm or the eggs will be haploid, which means they can only have 1 copy of the gene. That means they can have only 1 allele. That's confusing. Hop back to meiosis and refresh on that material. So when these chromosomes here undergo meiosis, the alleles that are going to be produced will either have a big T or a little t. You can't get a little t out of a big T genotype, and you can't get a big T out of a little t genotype. So Mendel revealed this by taking plants which were homozygous dominant in respect to the T gene and homozygous recessive in respect to the T gene and crossing them. And so that's a monohybrid cross. It's called a monohybrid cross because we're dealing with 1 gene, and we're hybridizing the 2 plants together. We're crossing them together, so that's called a monohybrid cross. So let's take a quick look at the chromosomes. Let me grab a pencil here. So in this genotype here, big T, we've got 2 homologous chromosomes, remember, 1 from mom and 1 from dad. So dad's going to be in blue, just because that's convention, whether it's right or wrong is incidental. And then the maternal chromosome is going to be in red. So remember, in a diploid organism, you have 1 chromosome from mom and 1 from dad. And so in the homozygous dominant plant, all the cells are going to have these pair of chromosomes, these homologous pairs of chromosomes, 1 from mom, 1 from dad, and one of those pairs of chromosomes will have this T gene on it, and there'll be a big T, and on the other chromosome there'll be a big T. So the homologous pair of chromosome is big T. So that's one plant. And then the other plant that Mendel was looking at, was going to cross, has the genotype that's homozygous recessive. And so again, we got this pair of homologous chromosomes, 1 from dad and 1 from mom, and both the chromosomes in the homologous pair have the recessive allele, so it's homozygous recessive. So Mendel crossed these two plants. And before we actually look at the outcome of the cross, we're going to think about what happens in meiosis, and then think about what happens at the point of fertilization. So let's just think about the big picture there for a moment. Meiosis is going to take these homologous chromosomes, and it's going to replicate them once, and then it will separate out the homologs, and then it will separate out the sisters. And so what we're going to do is produce gametes which have only one allele for each gene. And so for the plant on the left here, which is big T, the gametes can only have big T. There's no other variation of that allele for them to have. And on the right, that plant's going to have gametes with the variation little t. So let's take a look at how we represent that genetically. So when we do these crosses in simple Mendelian genetics, we create these structures called Punnett squares. And the Punnett square lets us make a prediction about the frequency of the genotypes in the offspring, and from that we can make some deductions about the frequency of the phenotypes in the offspring. So we're going to call the homozygous dominant and the homozygous recessive plants the parents. So this is the P generation that we're dealing with at the moment. So let's just put a P here to keep track of things. So when the two big T's on those homologous chromosomes get replicated, and then the homologs separate, and then the sisters separate, each gamete can only have a big T if we're dealing with a homozygous dominant parent. And the other type of gamete that could be produced is going to be big T. You can only make big T containing gametes, but they could have the chromosome from mom or the chromosome from dad. The same is true with the homozygous recessive plant. It can only produce gametes that are little t. Now, there are two types of gamete in that we could have a gamete with the maternal chromosome or with the paternal chromosome. So we're showing the full Punnett square here, but the outcome would be the same as if we just did a big T and a little t. So let's now think about what happens at the point of fertilization. So at the point of fertilization, we could bring a sperm, and sperm are produced by plants. They're contained within pollen grains. And so we could think about the sperm. So let's make this top row up here the sperm, that's dad, and let's make this column here, let's make these the female gametes, let's make this the eggs or the ova. So these are the sperm along the top, and these are the gametes, the ova down the left. So we can bring together a sperm and an egg and fuse them together, and what's happening there is we're bringing together this little t and this big T when we bring this sperm up here and this egg down here together. And all we're looking at is one gene here. Now bear in mind, there are thousands and thousands and thousands of genes in these pea plants, and we're only focusing on one of them. So the Punnett square only has this one T gene with our two alleles that we're dealing with. So when we do that, we're going to get an offspring, and let's think about what the generation would be here. This would be our F1 generation, right? So the F1 generation here would have the genotype heterozygous, and it would have a big T and a little t. And because we brought the big T in from the egg, from the sperm there, and the little t in from the top, we've got big T, little t, so we're heterozygous. And if you brought then this gamete together with this gamete, and this gamete together with this gamete, you should be able to work out all the possible combinations of the offspring in the F1. And so if you do that, is what you'll end up seeing is that everything's going to be the same. So everybody in the F1 is going to be heterozygous, big T, little t. And the only difference here is whether the cells of the offspring contain the chromosomes from mom and dad, or just from dad or mom. Now, we don't worry about that. All we care about is the combinations of alleles. So that's what's happening during meiosis. And so we set the Punnett square up in the first place, putting the gametes from the two parents along the top, and then down the side, and then when we bring the gametes together, you can think about that as being fertilization. And so you've got to fill all the Punnett squares in. So what you see here is that all of the offspring would be heterozygous. So we would say that 100% of the offspring would be heterozygous, and 100% of the offspring would have the phenotype of being tall. And so we make the deduction about 100% being tall from looking at the genotypes. So let's now kind of think about this from kind of a broader perspective and actually look to see what would happen if we did a second cross here. And this is where Mendels experiments become interesting. In that first experiment that I just showed you, that first cross, is what Mendel really showed is that you don't get blending of the effects of the alleles. So that first cross showed us that you have discrete effects of the alleles. So what that means is that when you bring a big T and a little t together, you'll get 100% tall plants. That means that these alleles, in this case, are responsible for producing discrete phenotypic effects. At the time, a lot of people had the idea that if you bring two alleles together, and obviously in Mendel's time, we didn't use the language of DNA and alleles and things, but we can do that now because it makes the discussion easier. At the time, people thought if you brought a big T and a little t together, that you would get something in between, and people thought that the effects of the two alleles would mix. And so Mendel's first experiment really showed that that didn't happen, that really one allele was dominant and one was recessive, and so those two things are very closely related. So let's think about what would happen if we did a second cross. So I've just put Mendel's first cross here in his first law. So that's his first cross where he crossed big T with little t, little t. Now the insightful thing about Mendel here, or the work that Mendel did with his gardener, was that he then took the F1 generation. Here is F1 generation, and remember, all of the F1 generation, 100%, are going to have the genotype of being heterozygous, and they're going to have the trait of being tall in the characteristic of height. And so both of these plants here are tall, and they've got the heterozygous genotype. So here's what Mendel did next. He took two of his plants that he got from his first cross, and he crossed them. So he took basically sperm from this plant, pollen from this plant, and fertilized this plant over here. And the good thing about these plants is actually you don't need to have two plants. You can take just one plant and you can do what's called self-fertilization. So you take the pollen from one flower and you put it on the female reproductive structures of another flower, and that works for certain types of plants, certain species of plant. So Mendel's taken the pollen from the plant on the left and the eggs from the plant on the right, and so he's basically done a cross-fertilization between these two plants. So in kind of genetic speak, we would say that he's crossed this heterozygote with this heterozygote. Now, if we think about what's going to happen in the flowers, the reproductive structures of this plant on the left here, if we do meiosis, the gametes can either have a big T or they can have a little t. Now they cannot have both, remember, because gametes have to be haploid, and so they can have one or other of the two alleles that are present. In this case, they can have a big T or a little t. And the same is true for the plant on the right. There can be a big T in the gamete or a little t. So now, if you think about the cross, if you think about taking this gamete and fertilizing this gamete, and you trace these lines together, here would be the genotype of the offspring. So if we put the big T in a sperm together with a big T in an egg, you end up with big T when you have fertilization. And so the offspring there would be tall. You wouldn't know, unless you knew what the genotypes of the gametes were, that that offspring was big T homozygous dominant. So when you do all of these fertilizations and you think about the offspring, what we find out is that in total, 3 of the offspring would be tall. But when we bring this recessive allele here together with this recessive allele here, we find one dwarf or one short offspring. So from the F1 generation cross, Mendel showed us that you get this ratio of 3 tall plants to 1 dwarf one. And now that we know about the genes and alleles, we can think about the genotypes. So the phenotypic ratio is 3 tall to 1 dwarf. And if we think about the genotypic ratio, we've got 1 homozygous dominant here, 2 heterozygous, and then 1 homozygous recessive. So Mendel's law of segregation has shown us that alleles have discrete effects on the phenotype, on the characteristic, producing tall or short in the case of plant height, and that in some cases one allele is completely dominant over another, and so the allele for determining tallness is dominant over the allele for causing shortness. And he showed us lastly that these alleles segregate in diploid organisms. That means they separate during meiosis. So that's what Mendel's law gives us. It's a pretty simple law, and I'll put some additional resources online so you can take a look at that. So that's Mendel's first law. We'll be back next time with Mendel's second law, which gets a little bit more complicated.